the open-ended section. There are 4 exams listed in the syllabus. You can find that on Canvas, when the dates are, and what chapters are going to be covered. The 4th exam is not a cumulative final exam. It's just a normal hourly exam like you're going to get in this class. But they are going to be given, the 4th exam is going to be given during our final exam period. As soon as I can figure out when that is, I'll be sure to put that into the syllabus. All right? So I'm still going over the syllabus, making sure that the dates are correct. On the schedule, right, on the list of classes, those dates are correct, right? So any other dates that you see elsewhere in the syllabus might not be exactly correct yet. I'm still going over and finalizing everything. But in that list of classes, in that list of dates are correct. So you can refer to that for when the exams are, again, except for the final. Homeworks, right? Each chapter is going to be assigned homework for the material that we cover. The homework is going to be due the midnight on the day before the exam that chapter is going to be covered, right? So if you're covering chapter 5 on exam 1, chapter 5 homework is due midnight the day before exam 1. Okay? These are meant to kind of resemble the kinds of questions you're going to see in the open-ended part of the exam, right? So they're going to be, show this or draw that type questions. What I'd like to do is at the beginning of each class, kind of open it up for about 10 minutes for you guys to ask questions about the homework. My advice would be not to leave it until the day before the exam. You're going to drive yourself crazy and you're not going to have enough time to study. And again, I want to open up the class about 10 minutes before the first 10 minutes of class for you guys to ask questions about the homework, so we can go over those together here. I don't care if you get the answers here, right? If we go through the problem and you just want to type in the answer, fine, right? But it'd be good for you to pay attention while we're doing these problems. If anyone has questions, some of the homework questions can be a little bit tricky or difficult. The quizzes, right? The last part, that's worth 15% of your grade. The quizzes, worth another 15% of your grade, are meant to be done before class, right? So they're due at 10 a.m. every day that we have a class meeting. What that means is that you are going to have to read the chapter before class, answer or take the quiz, and then come to class, right? Why are we doing this, right? Well, what I found, right, the question I always get 2 or 3 days before the exam is, how do I study for the exam? And I say, what are you doing? The student says, well, I just look at the slides. This is not an efficient way to study for the exam. You should be reading each chapter at least 2 times, once before class. Quizzes are meant to be addressing, and once after class, right? That will help you solidify everything that we go over, right? Fortunately, for you guys, that means you have to kind of try to learn on your own beforehand, and that will make your time in class a lot more productive. Instead of just me talking at you for 80 minutes, you'll be able to more efficiently follow along and understand what we're talking about. Right, so that's another 15% of your grade. Now these quizzes are kind of cool. They're called adaptive quizzes. So they kind of meet you where you're at, okay? If you are having trouble answering questions, the difficulty of the question goes down, right, until you start to answer them correctly. It'll start to ramp up until you get to something a little bit more difficult, so they kind of help to meet you where they're at. The last few years that we've tried this, the quizzes take about 10 to 15 minutes to complete, so it shouldn't be an onerous amount of work. In theory, right, everyone should be able to get 100% on all of the homework assignments and on all of the quizzes, right? So that's a free 30% of your grade right there, okay? The homework assignments have no penalty for how many times it takes you to answer, right? You can answer as many times as you want, right? As long as you get them correct, right? I have no interest in penalizing you for trying different things to get to where you need to be, all right? So you should be able to get 100% on all the homeworks, 100% on all the quizzes, okay? Aside from that, we have our exams, which we've already gone over. That's worth the remaining 70% of your grade. And that's it. So before we get into the actual biochemistry of it all, any questions about the class? No? All right. Okay, good. So let's go ahead and get started here. Chalkboard situation is unfortunate. I do like to use the chalkboard, but we'll have to see what we do. All right, so first of all, what is biochemistry? So biochemistry is, kind of cliche, the chemistry of blood, right? But what does that actually mean? Well, for the most part, what we talk about in biochemistry is the interplay between two different classes of molecules, macromolecules and metabolites. Now, macromolecules are typically biopolymers. So these things include proteins, which are a polymer made out of amino acids. They're DNA, a polymer made out of deoxyribonucleic acids. RNA, biopolymer made out of ribonucleic acids, right? These polymers, these biopolymers, assemble into sometimes gigantic molecules, molecular weights of millions of grams per mole, right? So these are truly humongous molecules, especially if you think about DNA. If you think about one strand of DNA, okay, well, now you're talking about hundreds of thousands of grams per mole. Now, we don't have a mole of DNA in each cell, right? Otherwise, we weigh hundreds of thousands of grams. But important to consider that these molecules are humongous. But they don't have to be, right? Some proteins can be as small as 50 amino acids, right? Which would be about, 50 times about 5 grams per mole, right? So not huge. But what is important is that these macromolecules almost always interact with some kind of metabolite. These metabolites are, for example, fuel molecules like carbohydrates, glucose, right? Glycerol, which is another type of metabolite. We're going to see glycerol when we talk about fatty acids or phospholipids. There are thousands of different types of metabolites. And these metabolites exist kind of in a steady state, right, in the cell. So when we talk about thousands of different metabolites, these are all more or less in the same cellular compartment. There are some exceptions, of course, when we talk about the mitochondria, for example, when we go over oxidative phosphorylation. But if we consider just, for example, glycolysis, which is using glucose to make energy, right? This involves 10 different reactions involving different proteins, different macromolecules interacting with different metabolites, different intermediates in that glycolytic pathway. All of these are interacting and existing in the same space. On top of that, right, when we look at, for example, glycolysis, it's very easy to put that into a box, right? I told you there's 10 steps, right? So we say, okay, glycolysis starts at glucose, and as we'll see, it ends at pyruvate, right? No big deal. What we have to remember is that all of these different pathways we're going to talk about, fatty acid oxidation, fatty acid synthesis, glycolysis, gluconeogenesis, glycogenesis, right? All of these different pathways exist together and they talk to each other. In fact, very often they share metabolites in between them. Right? This is how they can communicate and help to regulate each other. There are some pathways that we want to be turned on only when a certain pathway is turned off, or vice versa, or we want two pathways to be on. So all of these different pathways exist together, they talk together through the use of these metabolites. These metabolites can act as messengers in between these different pathways that we'll go over. Right, so here we have, for example, 3 different proteins, types of macromolecules. The important thing to consider about biochemistry is that for the most part, right, when we talk about proteins and their function, or any macromolecule and their function, what matters the most is not the composition, not the size. What matters the most is the shape of that macromolecule. These, for example, we're going to go over DNA today. DNA has a very specific structure and a very specific shape that allows it to do what it needs to do. In the case of DNA is to encode genetic information. In the case here, what we have is a binding protein, right? This is a specific type of DNA binding protein. And we see is that if we look at the shapes of the different proteins, we're going to go over protein structure on Thursday, but for now, let's just say, all right, this is how the proteins actually look, right? Don't get too concerned about what makes them up. But we see that they have roughly the same shape, right? They have 2 lobes here with a little crevice in the middle. Now, these binding proteins were isolated from 3 different organisms. We have one from humans, one from plants, and one from an extremophile. But all of these do the same thing. They're all binding proteins. They all bind DNA in the same way, even though they were taken from different organisms and are composed of different amino acids. Again, we'll go over structure on Thursday. We see that they end up having almost exactly the same shape.
because the job that they're doing is the same, they must have the same shape, right? In fact, when we look at evolutionary relationships between different organisms, the best way to do that is to look at how similar the shapes of the macromolecules in those organisms that you're comparing are, right? So we can see that humans and plants are more similar than humans and this extremophile. The shape here is much different, right? Well, not much different, but more different between humans and the extremophile versus the plant and the human, right? So we must be more closely related to the plant than we are to the extremophile. And that's how we start to put together these kind of phylogenetic trees, these evolutionary trees, right? Look like this. So we can see here, by examining the structure and the composition, right? So it's not only the structure, because we don't always have that information available, but by examining the similarities in the macromolecules and in the genetic information encoded in those macromolecules, right? We can start to put together these evolutionary trees, right? So we can see here archaea, eukarya, and bacteria. We see that we start to branch off, right? Archaea here, eukaryotes, like here we have yeast, here we have humans, right? We see that these are very closely related. How did we figure out that we are closely related? Well, by looking at the DNA sequences, looking at the structures of the different proteins, right? In fact, this is why yeast is used very often in laboratories as a model organism. It's easy to cultivate, it's a single-cell organism, but it's still a eukaryote that is closely related to humans, right? So they actually share a good number of the genes and a good number of the proteins that we do. Now, in this class, we're going to be concentrating mainly on bacteria, not because bacteria are especially interesting, but because it's easier to illustrate the concepts of biochemistry by examining bacteria, because they're just simpler. The principles that we go over in these bacterial examples can be applied to humans, right? And we're going to go through some of those. Now, before we get into all of this, right, looking at macromolecules, examining protein structures, seeing pathways, examining metabolism in detail, what I want to go over today is Chapter 1. Chapter 1 really just goes over some of the types of interactions that we're going to encounter, some of the thermodynamic principles that are going to be important to understand going forward. Right? We have to remember is that we're essentially just a walking bag of chemical reactions. So the laws of thermodynamics still govern everything that occurs in our bodies or in the cell of a bacteria. So to understand that, right, is crucial to understand how these different pathways are thermodynamically favorable, how they go forward, right, and how these different macromolecules interact with each other, right? Usually they don't interact using the covalent bond, and instead they usually almost always interact using weak interactions. We'll go over here. And DNA here just illustrates that very well. Now we're going to go over DNA more in chapter 8, I think, right? Later on, we're going to go over DNA in more detail. But for now, right, we just want to use DNA as an example to go over these thermodynamic principles and these weak force interactions. Right? So let's look at DNA structure first quickly here. So DNA is a polymer made up of monomers. In this case, the monomers are 2'-deoxyribonucleic acids. So a nucleic acid is made up of three things. We have our sugar base, right, which is here, this deoxyribose. So here we're missing a hydroxyl on the 2' carbon. Don't get too worried about the numbering here just yet. We're going to go over sugars in a lot more detail in chapter 11, so this will make more sense when we talk about sugars. But for now, just know that here, this pentagon here, this is the ribose sugar. What exactly a sugar is, we'll talk about in chapter 11. We have our base, right, our nitrogenous base. We can have one of 4 bases attached here to the carbon number 1 on our ribose. That could be adenine, thymine, cytosine, or guanine right here. Let's not get too bogged down with that just yet. But if we look at the rest of the carbons here, they're numbered as such, right? We have a 1', 2', 3', 4', and 5'. So these 5 carbons are all part of the deoxyribose sugar. Okay, now why are they called prime? Well, that's just to distinguish it from the atoms in the base. The atoms in the base are numbered without a prime, and just to distinguish the numbers on the deoxyribose are numbered with a prime. Each of the monomers are linked to the next monomer via this phosphodiester bond, right? So the last component of a nucleotide, or deoxynucleotide, is this phosphate here, right? So the 3' carbon of one sugar connects to a phosphate. This phosphate connects to the 5' carbon of the subsequent base, right? And what this means is that DNA actually has a polarity then. If we keep adding bases on and on and on, we see that one end has an exposed 5' carbon over here, and one end has an exposed 3' carbon over here. So we call this the 5' end, and we call this the 3' end. So DNA is not symmetrical, right? There's a 5' end and a 3' end, and when we talk about DNA, we always talk about it in the 5' to 3' direction, for reasons that we'll talk about when we talk about DNA. For now, just know that we always deal with DNA in the 5' to 3' direction. Now here are 4 bases: adenine, cytosine, guanine, and thymine. These are connected always via a nitrogen to the 1' carbon of our deoxyribose. So we can see that here, right? So we have a N-glycosidic linkage to the 1' carbon. What a glycosidic linkage is, again, we'll talk about in chapter 11, but for now, just know that the bases are always linked via a nitrogen to the 1' carbon of the deoxyribose. Now here, what I've highlighted is that we have sites for interaction. The thing to consider about the bases in this case is that they are mildly hydrophobic. We'll go over the hydrophobic force in just a minute. But they're also presenting this face here. This face is exposed to the environment, right? And this is important because here is where we're going to get interaction with the opposite strand of DNA. I'm sure everybody knows DNA is double-stranded. They form base pairs, and they form base pairs on these sides because these are the sides that are exposed, right? So those can be sites for interaction with other things, in most cases, another strand of DNA. Like we see here, right? So we have 2 strands of DNA. Notice, let's start with this one. Our 5' end is over here, so we have an exposed 5' phosphate over here. We come over here, on this side, we have our 3' end, right? So we have an exposed 3' carbon with a hydroxyl on it. The other strand, notice, goes the other way, right? So we start 5' over there, and we end with 3' over here. So this is called anti-parallel. The binding configuration of DNA is such that they are anti-parallel. One runs in one direction while the other runs in the other direction. Why does this matter? Well, it's only when we adopt this anti-parallel configuration that we can get correct base pairing in between the pairs, right? Let's go back here. Remember, we have exposed sites for interaction here. We need corresponding sites for interaction on the other strand. This can only be achieved when we have anti-parallel strands. So we can see here, for example, we might have an AT base pair. Over here, right, we might have a GC base pair, and they're held together by hydrogen bonds. Okay, so AT base pair has 2 hydrogen bonds, while a GC base pair has 3 hydrogen bonds. We're going to go over in more detail hydrogen bonds in just a second, but notice that here we have this configuration of hydrogen bonds. This can only occur when the strands are anti-parallel, right? If we try to base pair them parallel, so both running in the same direction, this would be down here, and this would be up here, and these hydrogen bonds wouldn't be able to form. And the same is true for the GC base pair, right? This one would still be able to form, but now this would be down here, and this would be up here, right? So we'd have scrambled our hydrogen bonds, so they cannot bind in a parallel configuration. They have to be anti-parallel. Additionally, an A will always pair with a T, and vice versa, a T always with A, and a G will always pair with a C, and vice versa. The reason for this is, again, what the identities of the groups that are exposed on this side. Notice here, this line is where it's connected to the deoxyribose down here. So this is the face that's exposed to the environment. The identities of these groups here, right, they need a corresponding interactor on the other side, right? This cannot base pair with this, right? It just can't. This could base pair with this, right? So, okay, so we can have an A base pairing with a G. This group here base pairing with this O. But, let's see, oh, this over here, this could also base pair with this H. So we could get an A-G base pair, okay? That is a thing that can occur. But why don't we, right? Well, again, we'll go over hydrogen bonds in just a second, but here, this hydrogen bond donor can interact with this hydrogen bond acceptor. This hydrogen bond acceptor can interact with this hydrogen bond donor. So you can imagine we took this, flipped it, put it over here, we could get these hydrogen bonds. The reason that we can't do that is that both adenine and guanine are purines. Purines have this bicyclic structure, right? Pyrimidines are monocyclic. So while, yes, the base pairing could work between A and G, because both are purines, it would be too big to fit inside of the double helix, right? So we always get a base pairing between a purine and a pyrimidine, never between two pyrimidines or two purines. The two purines are too small to fit inside, and the two pyrimidines, no, the two purines are too big to fit inside, the two pyrimidines are too small to fit inside. So again, we see that the structure of DNA is helping to accomplish its goal, which is to encode genetic information. We always want to know exactly what is base paired with what, right? And we want to make sure that that information is secure and is easily replicated. So here we have a very simplified schematic for how DNA is replicated, because we've already established that A can only pair with T because one's a pyrimidine and one's a purine, and they have corresponding hydrogen bond donors and acceptors. And the same is true for G and C. Well, now this gives us a little bit of an edge, because if we know the sequence of one strand, right, we immediately know the sequence of the corresponding strand, right? So by pulling the two strands apart, well, now we can synthesize a new strand, right? All the information for the other strand is already encoded on the parental strand. So we can end with two new double-stranded DNA. So why does this matter? Well, this illustrates again that the shape, the structure of DNA is uniquely suited to do what it needs to do, which again is to encode genetic information faithfully and to be easily replicated when the organism or cell divides. Here we see that we maintain base pairing by only being able to fit a purine and pyrimidine, never two purines or two pyrimidines, making sure that the hydrogen bond donors and receptors must always match, right, so that we enforce this pairing scheme. And this pairing scheme allows this process to occur, pulling the two strands apart and synthesizing a brand new strand. Because now we know that it's always the case that when there's a C in one strand, there must be a G in the other strand, and etc. Now, so now let's look at the different types of chemical principles, different types of interactions that we're going to encounter in this class through the lens of DNA, right? Just to be able to put an example to it, right? So let's start with the types of chemical bonds, covalent and non-covalent. Well, covalent, you know, that's the bond everyone knows and loves, right? This is when you have two atoms and you see a line in between them, like we have here. Right here, we have a covalent bond. What this means is the atoms are sharing a pair of electrons, right? Example of carbon-carbon bond, about 1.54 angstroms in length, right? And is very strong, okay? Here it's got a bond energy of 355 kilojoules per mole. In this class, we're going to be using kilojoules as our energy, right? Now, this is exceptionally strong, all right? And just to put some perspective on it, the hydrolysis of one ATP molecule releases about 30 kilojoules per mole, right? So this is about 12 ATPs worth of energy just in a carbon-carbon bond. It's not to say that they can't be broken. In fact, in all of the pathways that we cover, there are going to be some bonds that are broken, some bonds that are formed. But the important thing to consider is that because there's energy in that bond, it will take this amount of energy, 355 kilojoules per mole of carbon-carbon bonds, to break all of those bonds. 355 kilojoules is a tremendous amount of energy, right? Why or how then can these bonds be broken or formed, right? Well, that's why we're going over thermodynamics, and we're going to keep going back to thermodynamics all throughout the semester, right? There must be energy available in a reaction or in a system of reactions to do what you need to do or what you want to do. That is to break a carbon-carbon bond, then you need to find this amount of energy somewhere, right? Or couple it with another reaction that can provide that energy, right? We'll see examples of that. Double bonds, right? They're sharing two pairs of electrons. Now this is roughly twice the energy, right? So more electrons being shared, more energy is going to take to break that bond. Now the important thing about, or at least for us, covalent interactions is that they can exhibit a phenomenon called resonance, right? So here we have adenine, right? Here we have the structure of adenine. I showed it to you before. Over here, adenine. Now this is a bit of a lie, it's a bit of a simplification. This is true, right? But not quite. Because if we look, we can draw adenine either like this or like this. Notice the position of these double bonds has changed in the six-membered ring. Either one of these is correct. It satisfies the valences of all of the involved atoms, right? So that's fine. So either one of these is correct. So which one then exists in real life? Both. How can both exist in real life? Huh?
Aren't they both equally stable?
Yeah, they're both equally stable. So which one, if I were to pluck an adenine out of one of your cells right now, what would it look like?
It goes back and forth.
Right, goes back and forth, right? Almost instantaneously. So what actually exists is either this or this, but on any realistic time scale, what we see is that we have a superposition of both of them, so that each of these bonds exhibit partial double bond characteristics, right? Sometimes that's drawn with those dotted lines. We're going to see this again on Thursday when we talk about the peptide bond that holds proteins together. But when we do have structures like this, which we do often in these metabolites or macromolecules, when we do have this resonance, what does that mean for the molecule? Yes, of course, neither one is exactly correct, but what does it mean for a molecule if it exhibits resonance? Yeah, it's more stable. In fact, all of the different bases in DNA and RNA exhibit resonance, which is good news because what do we need DNA to do? Well, to faithfully encode genetic information. So the more stable we can make the molecules making up DNA, the better it's going to be able to do its job. Right? So here, it's just an example of adenine, but all of the bases exhibit resonance. They're all more stable than, say, if this were a carbon, right? Because that wouldn't be able to exhibit resonance, or as much resonance. But more interesting than the covalent interactions, right, is really the non-covalent interaction. And we have four different types: ionic, hydrogen bonds, van der Waals, and hydrophobic interaction, which is not really an interaction, it's kind of an illusion, as we'll see. But let's cover ionic interactions first. So ionic interactions are easy to understand, right? Opposites attract equals repel, right? So if you have two magnets, one is north, one is south, they come together and stick. If you have two norths together, they repel. Two souths together, they repel. The same is true for charged particles, right? So if we have a positive and negative charge, they're going to be attracted together. If we have two negatives or two positives, they're going to be repelled. And we can quantify the energy in that either repulsion or attraction using this equation right here, right? The energy. So the energy in a ionic interaction is equal to K, Q1, Q2 over dr. So let's go over each of these terms. K is just our proportionality constant. That's just a truth of the universe. For our case, because we're using kilojoules per mole, that's 1,389, right? 1,389 kilojoule angstroms per mole. Q1 and Q2 are the charges of our two particles. So here we have particle 1, particle 2. Let's say that this has a +1 charge and this has a -1 charge. right? So we just plug those in here. These charges are not in coulombs, they're in units of electronic charge. So for example, we have a sodium and a chlorine. Well, sodium is Na plus and chlorine is Cl minus. So we don't have to do any conversion between, we have one extra electron, how many coulombs is that, right? Well, this is units of electronic charge, so it makes our lives very easy. The sodium would have a Q of +1 and the chlorine would have a Q of -1. Plug those in. That's part of what the proportionality constant allows us to do. Divided by dr. D is our dielectric constant. This is unitless. And for water, which all of our reactions will take place in, that's 80, right? What this means is, right, we consider we have the Q1 and Q2 just out in outer space or in a vacuum, a perfect vacuum. Well, the dielectric constant there is just 1. There's nothing getting in the way of those two particles either being attracted or being repelled from each other. Once we move it into a medium, well, now that medium is going to begin to interact with those particles as well, right? So, how do we take that into account? With the dielectric constant. That means that we have the same two charged particles in space. They will be attracted 80 times stronger than they are as if we do it in water, because that water is attenuating some of that force, right? It's interacting with the particles. So for water, that dielectric constant is always 80, and R, which stands for radius in this case, is really just the distance between the two particles, but importantly in angstroms. Okay, that has to be in angstroms, not nanometers or micrometers. It must be an angstrom. Again, that's where the proportionality comes in, right? It's assuming that the radius is in angstroms. Well, now if we plug all of this in, that gives us an energy in kilojoules per mole, right? And if we consider, again, let's say that we have a +1 charge and a -1 charge here. Let's just ignore all of the different things here. Let's say that they're all 1, okay? Well, now our E is +1 times -1, so that's -1. Well, let's think about what's going to happen when we have a plus and a minus charged particle close to each other. They're going to be attracted or repelled? Attracted. And is our E negative or positive in this case? Negative, right? So when E is, yes, less than 0, we have an attractive force. If E is greater than 0, then we have a repulsive force. Let's again consider if we have a +1 charge next to a +1 charge. Well, now E is K, which is positive, times +1 times +1, so that's still positive, divided by 80, which is positive, divided by R, which must be positive, so E is going to be positive. So if E is greater than 0, then we know that we have a repulsive force. And if E is less than 0, then we have a attractive force. What if E is 0? How could we have an energy of 0? If one of the charges is 0, exactly, right? So here, let's say that Q1 is +1 and Q2 is 0, okay? Well, then now our E is simply 0. This ionic interaction force can only take place if we have 2 charged particles. If one of them is not charged, and it's not even worth going through, because everything's going to be multiplied by 0 anyway. Let's move on now to, so how does this apply to DNA? Sorry. Well, if we go back to the structure of DNA, we see that down here, right? You notice that we have, yes, all the bases. None of the bases have a charge, so we can ignore that. We have all the deoxyribose sugars. They don't have a charge. We can ignore that. But notice each of the phosphates that sit in between the sugars on what we call the backbone has a negative charge. So each of the phosphates has a negative charge. Well, okay. Each of these phosphates then are repelling each other, right? But that doesn't matter because they're all covalently linked via the deoxyribose sugar, so that's not going to mean anything. However, remember we have an anti-parallel DNA strand that's being held only by hydrogen bonds to the original strand. And this second strand, the anti-parallel strand, will also have sugar phosphate backbone. So now let's just look at this phosphate here. This phosphate is going to be interacting with this phosphate on this backbone, this one, this one, this one, this one, and so on, right? Here we have strand 1, here we have strand 2, just make it a little bit easier to see. Now we have this negatively charged phosphate here. What type of interaction is going to be taking place with all of these other phosphates on the other strands? It's going to be attractive or repulsive? repulsive, right? They're both negatively charged. -1 times -1 is +1. So E is positive, means that it's a repulsive force. So this is just for one phosphate here. But we have to remember this is also interacting with all of these. This is interacting with all of these. This is interacting with all of these. This is interacting with all of these. So how do we prevent the DNA strands from being just pushed apart by all of these repulsive interactions between the phosphates on the backbone? What could we do, right? So don't try to think of what's the actual answer, right? But what do we want to do in this case? We have an E that's greater than 0. What kind of E would be the best? Huh?
One is the opposite.
Okay, sure. Could be the opposite, or 0, right? Could be 0 or the opposite. Now, changing the charge here is not quite an option, right? So ideally, yes, you are correct. We would want it to be an E less than 0 to hold them together. This presents a problem though, because now these will be attracted, right? What if that's enough to overcome the hydrogen bonds here? So now we're just turning the DNA inside out. But if we have it close to 0, now we can ignore this, and the only thing that will be holding the two strands together, as we intend, is the hydrogen bond interaction between the bases. So how can we do this? Well, we can't alter the phosphate, right? We can't alter the phosphate. Now we could try to protonate the phosphate, right, in order to get rid of that, but that involves lowering the pH significantly. We always want to keep the pH around 7.0 for reasons that we'll see later in the semester. So protonating the phosphate to get rid of the negative charge is not an option. Instead, what we do, or what the body does, is essentially coat the DNA with salt, right? Let's say that we have a mixture of DNA, we add some sodium chloride or some magnesium chloride to it, that ionizes into Mg2 plus and Cl minus, or Na plus and Cl minus. The Cl minus is going to be repelled from all these, right? Because of the same charge. Cl minus is going to be repelled from the negatively charged phosphates, so those just go off and we can ignore them. However, these are going to be attracted to the Na plus or Mg2 plus. So now, when we examine this region of space, right, not the phosphate in particular, but just this region of space, well, these two kind of cancel out, right? So now we have what's functionally 0 charge over here, or 0 charge over here, and this results in fewer ionic repulsions between the two strands, and in fact, this is the case, right? If there is not enough salt in solution with DNA, it is much easier to denature that DNA. In other words, to split the two corresponding strands apart. We need sufficient salt in our bodies and even in the lab to make sure that DNA remains double-stranded, right? So salt is actually quite important.
Just this whole NA.
Huh?
Maybe like another salt or just.
Could be another salt. Here's Na, here I saw I put Mg, could be potassium, right? Yeah, could be other salts. In fact, it often is. Now, hydrogen bonds, right? So we've kind of been dancing around hydrogen bonds here. I remember when I was so long ago to admit trying to learn hydrogen bonds in organic chemistry. It took me a while. But here we see that a hydrogen bond donor, or a hydrogen bond really is made up of two parts. We have a hydrogen bond donor, this is where the hydrogen is, and we have a hydrogen bond acceptor, right? So what creates a hydrogen bond is simply that the hydrogen on the hydrogen bond donor must be covalently linked to some electronegative atom, right? It just has to be highly electronegative. Now in our case, that's going to almost always be either a nitrogen or an oxygen, right? And why does this matter? Well, because hydrogen is not electronegative. If it's bound covalently to an electronegative atom, this steals electron density away from the hydrogen, giving this nitrogen or oxygen a partial negative charge, leaving the hydrogen with a partial positive charge, right? So there we have our hydrogen bond donor. Our hydrogen bond acceptor is simply another electronegative atom. This could be on another molecule. It could be on a different part of the same macromolecule. What matters is that it's electronegative. So whatever this is connected to, this is always going to be partially negative, okay? So now we have a partially negative next to a partially positive. This interaction here is our hydrogen bond, okay? So let's look at the picture. so we go back here to our base pairs, right? So now it's clearer to see. So here, for example, we have an NH, so we have an H bound to N. So here's our hydrogen bond, what, donor or acceptor? Donor, yeah. So here we have a partial negative on the nitrogen, a partial positive on the hydrogen. This is in close proximity with this oxygen here. This oxygen is our hydrogen bond, what, donor or acceptor? Acceptor, right? So here we have a partial negative. So this partial positive next to the partial negative gives us the attractive force that we see here, and the same is true down here, and the same over here, right? Here and here. Now, what do you see in all of these hydrogen bonds that are the same, right? They all, what? I mean, yes, they all have a hydrogen bond donor and acceptor. What else do they have in common? All right, but remember, hydrogen bond donors or hydrogen bonds don't care if this is a purine or a pyrimidine. No. All the hydrogen bond cares about is that there's a donor and an acceptor.
They're binding to the nitrogen.
Huh?
They're binding to the nitrogen.
Okay, so in this case, by happenstance, all of the hydrogen bond donors are hydrogen bound to nitrogens, right? But that's coincidence, right? What else? What else do they?
Acceptors never have double bonds.
The acceptors have double bonds.